Okay, then hello. Are there people in chat? It's so quiet out there in chat land. Hello, hello. Is anybody there? Yes. Okay. Good. We have our regulars in the chat. Good. 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 Okay. Here we go. So pyrimidine biosynthesis. Let's first talk about pyrimidine catabolism, right? So first off, all right, there are purines and there are pyrimidines, right? So we learned about purines last time. And this time we're going to learn about pyrimidine. So this is cytosine, uracil, thymidine, right? And as you remember from your basic biochemistry, thymidine is only found in DNA and uracil is found in RNA, right? But they serve the similar functions, right? So these are single ring bases. And then they get this different spinach that gives them different functionalities. Does this all seem familiar from your regular biochemistry chat? All is this known? Yes, good. So, so normally DNA is not degraded in cells, right? So that's should be fairly obvious. Degrading DNA is, unless they're undergoing apoptosis, is not normal. You don't normally break down your DNA. But RNA, on the other hand, is constantly turned over, right? So we'll talk about the catabolic products that come out of these processes where cytosine and uracil are converted into beta alanine, and thymidine is made into 3-aminobutyric acid. So we'll talk about these pathways here next. So one of the things that's great about pyrimidine catabolism is it follows something very similar to what we saw in purine catabolism. Okay, so first off, reminder on nomenclature. So when we have the base, the ribose, and a phosphate, we call it a nucleotide. And when you have just the base and the ribose, then we call it a nucleoside, right? And then when you just have the base, you call it a base, right? It's all about that base. So the nucleotidases, not surprisingly, are the ones that take the NMPs. So this is N is any nucleotide. And then they convert them into nucleosides and inorganic phosphates. And that's done through hydrolysis, right? So that's the same thing as the purine catabolism. So the uridine phosphorylase reaction then is the one that takes pyrimidine nucleosides and then converts them into pyrimidine bases. And then the cysteine deaminases are the ones that take this, cytidine and turning it into uridine and ammonium. Okay, so these are these different steps here that we're going to be looking more at the roadmap. So for pyrimidine nucleotide degradation, here are the pathways. They come in here as cytosine, and that can be converted into uracil via the cytosine deaminase. And then there's this these regular pathways that'll take it and ultimately convert them into the final product. So uracil goes through this oxidation and uridine phosphorylase catalyzes reaction for all bases. Uridine phosphorylase reaction. We will get through all of the reactions here one by one here, Brandon, in a sec. So the anyway, so the dihydropyrimidine dihydrogenase reaction is the one that converts the uracil into this intermediate dihydrouracil, and that one's ultimately converted into this funky intermediate, this uridopropanoate in carbamyl beta alanine molecule, and that's ultimately broken down into this beta alanine. So same thing here that we've seen in our other pathways. I want you to know the beginning point, the end point, and the major cofactors. So in this case, that you've got to reduce the uracil base in order to get it into a form that's usable, right? So you dump an NADPH in. Thymidine follows a really similar pathway. So it's also using the same enzyme, this dihydropyrimidine dehydrogenase. It's also reducing thymidine into this dihydrothymidine molecule. And it has a different end product here, which is this beta amino isobutyrate acid. So beta alanine is the endpoint from this degradation pathway. You probably recognize this from our supplements lecture, that beta alanine is also something that that some people take for sport supplementation. I'm not exactly sure what the rationale for that is, but it's like, I don't know, it's like eating uracil poop, make you stronger. And then there's also beta aminobutyrate. If you find it, that it can be an indicator of DNA turnover. Okay, so the precursors of pyrimidines are orotate. And then that's also ultimately what you use to make thymidine, cysteine, and uracil. So we'll talk about how we go from there. Orotate is, did you guys in your, I can't remember, we were considering this when we were prepping the tutorial section. Did you guys end up looking at dihydroorotate and orotate in your malaria lectures? Or in your malaria tutorials?
They're looking at that this week.
They are this week. Okay. So it's this, yeah, it's okay, cool. So I have a long history of science with these pathways. Dihydroorotate dehydrogenase is a famous enzyme in malariology. You know, it's one that's critical for the parasites. And weirdly, it appears to be, so it's, you know, it's this rate controlling step in making pyrimidines, and it's a very druggable target. And so, you know, it's been researched for a long time. But one of the things that's peculiar about dihydroorotate dehydrogenase is that, as it turns out, you can, you know, it's totally dependent on the function of the electron transport chain in malaria. And so, you know, it's directly embedded into the electron transport chain, but in yeast, they can get a cytosolic version of the enzyme. And so as a result, there have been, you know, examples where you can actually decouple the electron transport chain from it by using a yeast dehydrogenase. Anyway, there's a very long, interesting history of studies of yeast metabolic function, or I'm sorry, of malaria metabolic function in this enzyme. It's a really important enzyme. It's very druggable. It's also very druggable for cancer and some other things. We'll talk about that. Okay, so how do you make it? This is an overview. So there's 6 steps from bicarbonate to UMP. So bicarbonate and ammonium becomes carbonyl phosphate. So we already learned about that before in our urea cycle section, but this is a separate CPT enzyme that we'll be talking about. And that makes carbonyl phosphate, which ultimately goes in and makes part of the ring. And then the rest of the ring comes from an aspartate. Then we use PRPP, which is this activated ribose, just the same we did in purine biosynthesis to help make the synthesized bases. So we have two atoms that ultimately come from carbonyl phosphate, 4 atoms that come from aspartate, and then a ribose that comes, you know, obviously from the PPP. So that's the overview. Well, let's talk about each of the individual steps. So de novo pyrimidine biosynthesis pathway goes like this. So you take this bicarbonate and you take an amine from a glutamine, the side chain from the glutamine, and 2 ATPs to make this carbonyl phosphate. So this is exactly the same reaction we saw in starting out the urea cycle that we were talking about before, except this is a separate enzyme here, CPS2, carbonyl phosphate synthase 2, and this makes this activated carbon molecule that you can use for doing biosynthetic reactions. So then from that, you add a aspartate, and this ultimately makes this carbonyl aspartate intermediate here, this guy. And so this is very important intermediate. This is like very first part of this, the committed step towards pyrimidine biosynthesis here. And this gets you into the pathway. So then dihydroorotate dehydrogenase, this is the enzyme I was just talking about before, it has this really important step where it makes dihydroorotate. I think, as Carla was mentioning, you're actually seeing these in your tutorials right now this week. So these are really cool molecules. Super important step. This that's dihydroorotate, and then it goes from dihydroorotate to orotate. Sorry, this is the step we're talking about, and it's dumping enzymes, electron equivalents directly into the electron transport chain, directly onto coenzyme Q. In the case of malaria, at least. Yeast can have a cytosolic version that uses NADPH, so there can be a few different electron acceptors there. Then orotate's then converted into orotate 5-monophosphate, or OMP. So this is a nucleotide that's not part of our standard nucleotide language. And then it's decarboxylated in the step to ultimately make uridine 5-phosphate, UMP. If you want to play around with the data sets that you've received then from Maven, like you can actually go and look at all these pathways get labeled in the data sets that you guys are working with. These are all data that I collected when I was at Princeton and studying these pathways. So you'll find isotope labeling down these pathways. It's pretty interesting. Okay, so after you have UMP, then UMP can be made into UDP, UTP, and then that's converted into the downstream enzymes. So we'll talk about those biosynthetic pathways. So, and that's done by UTP kinase, and we'll talk more about these things here a bit. Okay, so in detail, bicarbonate is not reactive by itself. It's quite happy to sit around and be in solution. And so to make it active, we use this carbonyl phosphate molecule, as we mentioned, and we use an ammonium from glutamine, ultimately making glutamine, and that ultimately makes this carbonyl phosphate. All these reactions that I'm showing here are done by this enzyme, this carbonyl phosphate synthetase 2, okay? And then you got to remember that CPS2 is not CPS1. If we were going to have a final exam this year, I would be making lots and lots of points to remember that, because that's a really great exam question, like being able to differentiate these. And they should be different because, they're regulating totally different pathways, right? Even though they result in some, the same intermediate, they're supporting two separate pathways. All right, so this is the same chemistry that we saw before. So then this next step that we talked about, aspartate transcarbamylase, where aspartate comes in, and that's providing most of the carbon chain then for the base, and it ends up with this carbonyl aspartate moiety. If you look for carbonyl aspartate, you'll also find that in your labeling data sets from malaria. So you can actually find and prove all these things really exist in the real world, not just on these lecture slides. And then dihydroorotase takes that carbonyl aspartate, cyclizes the rings, and you get a loss of water in that reaction with ring closing. And then that leads us to this really important reaction, dihydroorotate dehydrogenase. And that dumps it directly on coenzyme Q. Again, like they're different creatures have different electron acceptors. So we're showing this as the malaria version. I don't remember now what the humans use as an electron acceptor, but I know yeast uses NADH or NADH, I think, as a cofactor. Oh, it's right there on the slide. Class 2 is bound to the inner membrane. Class 1 is cytosolic. See, I just need to look at my slides. And then it goes through this intermediate called FMN. You'll see this in in a number of reactions that that interact directly with coenzyme Q, is it's mediated through this FMN flavin mononucleotide cofactor intermediate. So this is also a vitamin. So if you're ever wondering why flavin is one of your nucleotide or one of your vitamins, is because yeah, it intermediates these electronic transfers between coenzyme Q and your products. Anyway, allows us to make a arotate. So once you have a arotate, you're basically there. You just need to add it onto a ribose base. So arotate plus PRPP ultimately makes this rotatidyl this arotatidylate and pyrophosphate. And then here we are at this last final step where we lose carbon dioxide, and that makes this final molecule that is one step away from UTP, right? So so then the nucleotides, how we make them is, so we start with these monophosphates and we add on extra phosphates to make NDPs or nucleotide triphosphates. We do that with these either nucleotide monophosphate kinases or nucleotide diphosphate kinases. And then from UTP as a precursor, then we can make CTP. So it's important to remember this actually starts as a UTP as the biosynthetic start point for this. And cytadidyl synthase allows a transfer of an ammonium from the side chain of glutamine on with an ATP, and that aminates this right here, this section right here. So pyrimidine biosynthesis is really tightly regulated. This is a very expensive pathway to make these molecules, and so you need a fairly tight regulation to keep the cells happy. So this is done in a number of steps. One is UTP and UDP, which are the final end products here, are they're inhibitors of the CPS2 reaction. The transferase doesn't need PLP. Yeah, it doesn't in that case, does it? Huh. You're talking about this reaction here, Jeremiah? That's a amidotransferase reaction instead of aminotransferase reaction. I can't remember now, Carly, do those amidotransferase reactions use PLP as a cofactor? All the aminotransferases ones do.
No, the amido ones are different. I would refer to the asparagine synthetase reaction for like an example of amidotransferase.
Yeah, I don't think that they do use PLP. Actually, Jeremiah, if you want to hang on at the end of the lecture, I'll show you how you can actually research those questions specifically using KEGG. We just started to do that yesterday, and I can show you how I look up those kinds of details when you don't know from the literature. I don't think that it uses PLP though for amidotransfer reactions. So and then so obviously ATP is also feed forward stimulator of the pathway. So when you have energy, you can do it. When you don't, you repress, right? So you don't want to make nucleotides when you have no energy around. And then there's also some activation by PRPP. So when you have activated phosphate on a PRPP is a pyrophosphate ribose that allows you to, or not pyrophosphate, but phosphoribophosphate. It's got two phosphates on it. That's the one that allows you to continue putting the sugar onto the bases. And then there's some feedback inhibition for UMP onto in this transition from OMP to UMP. And anyway, so that's how the overall pathway is regulated. Let's see. So some other notes on here. ATP cases are the main regulatory enzyme in bacteria. ATP activates it, and CTP and E. coli and or UMP are most bacteria are inhibitors. So we'll talk a little bit more about how these, there's a very complex regulatory network here that we'll talk about how to ensure you get the right quantities of all the bases. Ribonucleotide reductase is a really critical regulatory element on ensuring you get all the right kinds of bases. As you imagine, it's fairly, this is for making deoxyribonucleotides, I should say. And so what you do if you want to make DNA, you start with RNA moieties, the nucleotides for RNA, all right, and you convert them into the nucleotides that you use for the deoxynucleotides. And you have a whole bunch of them: deoxy-ADP, deoxy-GDP, deoxy-CDP, and deoxy-UMP. And then this enzyme that does this ribonucleotide reductase, is this critical regulatory element. It's got this really beautiful cascading progressive regulatory mechanism to ensure that you get the right quantities of all those deoxyribonucleotides for making ATP. So first off, so what's going on here? Ribonucleotide reductase ultimately has to reduce this carbon 2 of the ribose, right, to get to make your deoxyribonucleotides. So it has two separate pathways for doing that. You can and we'll talk about those both. One is a pathway that uses glutaredoxin, the other one is one that uses thyroidoxin. So we'll talk about that here. Next slide. The pathway 1 uses glutaredoxin, which is, got this disulfide on it that enables the chemistry, and ultimately it takes this nucleotide diphosphate and this hydride from glutaredoxin, and that allows you to make a water and a deoxyribonucleotide, so the reduced nucleotide. And then that has to get regenerated, and the pathway 1 gets regenerated through glutathione redoxin or reductase, and with a NADPH as a cofactor. Coming from the PPP. So the pathway 2 uses a different redox source as thyroidoxin, and works similarly, except for ultimately FADH2 is the cofactor that will allow you to regenerate the redoxin that's driven by thyroidoctase. And ultimately that can be reducing equivalents from nucleotides like NADPH. So this is that really cool regulatory mechanism I was telling you about for ribonucleotide reductase. So you have all these pools of deoxyribonucleotides that you have to make in order to get uniform quantities of everything. And so how do you make sure that you got the right amount of each nucleotide? It's pretty complicated, but you need the same amount for each. So the way they do this is ribonucleotide reductase actually has all these subunits where the nucleotides combine. So what happens is that the final end product from the whole biosynthetic pathways is deoxy-ATP. And this guy, if you got too much of this, will shut down the conversion of all the other steps. But so there's this progressive action in chemistry that starts with CDP, ultimately making this deoxy-CDP, and the deoxy-CDP is one of your products. But the other one is that you get this uridine diphosphate, and this makes this deoxyuridine diphosphate molecule. And as we just saw, that's ultimately converted into this. You have this progressive action into thymidine. So each of these products is then used to inhibit the biosynthetic pathways of the receptive of the other steps, so that this sort of progressive regulatory actions ensures, like, so TTP is used to regulate this conversion of GDP into deoxy-GDP, and deoxy GTP is used to inhibit the production of deoxy-ATP, and you remember that deoxy-ATP was used to regulate CDP. So it's this beautiful sort of like interwoven regulatory network mesh that allows you to get the right balanced levels of all the deoxynucleotides. Cool. So formation of deoxy TMP. So this this is one of my favorite pathways here. So you take deoxy CTP and and you get rid of an ammonium here and this cysteine deaminase, and that makes the molecule dump deoxyuridine triphosphate. And since I'm juvenile, I find that really amusing. So, oh, sorry, that's deoxyuridine triphosphate. And so you have a pyrophosphate, pyrophosphatase that cleaves off 2-phosphate to make this deoxyuridine monophosphate, sorry, dump molecule, moiety. And so that is the required starting point for making this deoxy thymidine monophosphate. So you make dump, and that's then used as the precursor for thymidine synthase, using a methyl that comes from tetrahydrofolate. So methyl tetrahydrofolate is then allows you to make that thymidine monophosphate. And so this has to be regenerated. So after we make the thymidine synthase makes this, puts the methyl moiety there on the here on the side chain, and then you have to, and that's coming from your methyl tetrahydrofolate here, and so that ultimately has to get regenerated, and so that's regenerated here through this dihydrofolate reductase reaction, DHFR. This is also another drug target. Really important drug target, also drug target in malaria, DHFR is, and also target for cancer stuff and quite a number of other things. So a really important enzyme in enabling biochemistry of growing cells. So these reactions are all unique to growing cells, right? Because they're deoxy ribonucleotides. So there's this two enzyme step here through serine hydroxymethyltransferase, which is what is, your methyl group is ultimately coming from serine and regenerating glycine in the process, and then DHFR is the one that's providing the reducing power, so taking NADPH and reducing the DHF into tetrahydrofolate. Yeah, makes sense? From dihydrofolate to tetrahydrofolate. Is everybody following that so far? You need a reduced methylated tetrahydrofolate to use as a precursor, and so you have a 2-enzyme step that converts it from the end product into that which is dihydrofolate back into that methyl tetrahydrofolate. Yeah? Chat is very quiet today. Would we prefer to talk about French literature? Very quiet. Is there anybody in the chat? Oh, okay. No, that's gonna be a horror show if I do the French literature lectures. Okay, so these are really important, as I said, for rapidly growing cells, and so not surprisingly, are also targets for drugs. So AZT, which is the drug that's used for treating AIDS or HIV, is a mimic here of this thymidine nucleoside. And if you see something that's really interesting about it, like chat, what do you think, how does this enzyme work? Or I'm sorry, this inhibitor work? What does it do? Look at this crazy reactive azide here at the end. So this is the AZT is a azeto group. So this is an azeto moiety. Yeah, so right, this is called a suicide inhibitor. So Frida is right. So what happens is that enzymes like this get into get in there, and then they covalently bond themselves to the enzyme, and then the enzyme cease working. See, we've replaced this hydroxy here, which is coordinating the active site with this guy, and so that prevents them from working. So when I went to graduate school, or I was interviewing for graduate schools, one of the guys I was interviewing with, he asked me to draw the structure of the AZT on my graduate school interview, which now in retrospect was kind of like, I don't know why he would have thought I should have been able to draw it. But if you ever ask that question, here it is. That's the structure. Anyway, and so this is used for treating HIV. What does it do? It prevents the it prevents the virus from replicating, right? Next slide. Oh, shoot, we're done. Darn. So we talked about purine catabolism, de novo purine biosynthesis, regulation of biosynthesis, the thymidine monophosphate biosynthesis, pyrimidine analogs, and we talked about how making deoxynucleotides. And spoke briefly about how some of these things can be used as drug targets. We'll have quite a bit more about things related to this in our next lecture. We're going to talk about cancer, and nucleotide metabolism is very important in cancer, so we'll be talking more about that. If they're happy to take questions, if there are no other questions, I'm going to show again, which we showed briefly at the end of the class last time, but how you actually can search for the primary literature for reactions and that sort of thing now. Or how do people feel? Are you ready for a tour of KEGG?